First of all, I want to say thank you for the applause and thank you for coming. Most importantly, I really enjoy doing this for SEAC. I was at the University of Arizona and College of Medicine for 40 years, and so this is now my way of paying back to the community. But today we have a special seminar. Each of our seminars is special in its own right, but today we're going to hear about a unique, absolutely unique enterprise that happens to be part of the University of Arizona. It happens to be just outside the city limits of Tucson and Oracle, and it's the system called Biosphere 2. John Adams, our speaker today, is the deputy director of this unit, and he has been holding it together, and I mean in many, many different ways, from funding, from organizing research activities, from taking care of all the people who work there, and most importantly, perhaps, taking care of the 5 biomes, which constitute a kind of a miniaturized Earth. I think it's an incredibly interesting place. If you haven't had a chance to go visit, you really should. And today, John Adams is going to tell us, if you go there, sometimes even on tours, they take you behind the scenes, so to speak, and you go down in the bowels of the structure, and you see all of the infrastructure that supports the plants and the sea biome and so on. But today, John Adams is going to tell us about the research infrastructure that typically we don't hear very much about. So, John, it's all yours, and thank you for being here.
No, thank you, Rafi. Thank you, Gene, and we'll sign you up as our promoter here in a little bit, all right? Welcome, everyone. Thank you. It's great to be here. So I think the first way I want to get it started off, I think most everybody lives here in the Tucson area. Is that right? And so how many of you have been to Biosphere 2? Okay, so about half the audience, and I don't know who's out there on the web who's been there, but has it been recently or has it been a while ago? A while ago? You've been there recently, I know. Okay, so a lot has changed, but what I want to do for those of you who maybe don't know a lot about Biosphere or maybe have heard bits and pieces about it is start off with a little bit of history and sort of how we got to where we are today. Talk about that original group, talk about some of the interesting research that came out as it transitioned from the original private venture to a private college or university in the city of New York, Columbia University, and then talk about the University of Arizona's involvement and how we've moved this facility from one where a lot of people lived inside and there's tremendous amounts of publicity around that and hype to one that, you know, is a world-class research center today. So I think we've all seen this image in one way or another, and I think, you know, the Biospherians 25 years ago, because this year is the 25-year anniversary of it, were asking the exact same questions that they were actually doing. What if we could travel into space, and what would it take if we were going to colonize another planet? A lot of things that happen here at SEAC are somewhat around that central theme. We've got a lunar greenhouse up there that Gene works with and his team. And the notion is if we were to travel to another planet, could we grow plants, and what would it take to do that? But 25 years ago, you know, there was a resurgence in the space industry after we had landed on the moon, but no one knew what it would actually take to do something like that. So a group of folks were able to get together some funding to look at, you know, if we were to grow plants in a soil media and we were going to balance an atmosphere, what would it take? What would it look like? Could we even do that? And the notion is, you know, could we replicate some of Earth's systems? Because the idea was chemistry can get us, you know, part of the way there, but at some point, those biological systems are going to become vital. And the only place that we know that we've got those is right here on Earth. And how are we going to replicate those systems? So in the late 80s, early 90s, they began construction on this large glass structure. And originally, the structure was privately funded by a gentleman by the name of Ed Bass. The initial budget was around $35 million. to $50 million were the numbers that were put out there. Today we've gotten confirmation, and they ran just a little bit over, that it cost about $250 million. Okay, and so that was, if you figure late 80s, early 90 dollars, if we had to reconstruct a facility like this today, we're probably looking at close to $1 billion to do something like that. And in today's economic climate, the likelihood that you're going to have somebody that's going to finance that, whether it's a government, an agency, a private individual, is at least unlikely currently, or at least that's my opinion. So if you've not been out there, a lot of people ask, well, why did they select the site that they did to build this little over 3-acre structure? And first and foremost, I think the real estate was available for them at the time. But also, you think about our climate here in southern Arizona. So, if you're going to grow plants inside of a building, we can either supplementally light it, or we're going to leverage the natural light. What a great place to be here in southern Arizona with all of the sunny days that we have. Geologically, we're really stable. So you're going to build a big glass house. You don't want to have to deal with a lot of earthquakes. And, we're not the Midwest where you get a number of violent storms, large hail, for example, that also could compromise your structure. So as a result, that was one of their choices. And again, this gives, for folks who haven't been out there, some different perspectives of the facility and how massive it is. Again, that structure covers a little over 3 acres. Its north-south access is almost 3 football fields in length. And the approximate volume inside is about 7.3 million cubic feet. And when they built this structure, it was a facility that was one of the most tightly sealed buildings ever constructed. So of that 7.3 million cubic feet, it only lost about 10% of its volume annually. And the way they achieved that is, if you can imagine, it looks very different today if you were to go out there, and a lot of this would, but that underneath that foundation, they excavated, laid in concrete, and then they took stainless steel sheets and welded them together to create a stainless steel bathtub. They then built the structure and the infrastructure on top of that, and then the overhead is glass panes that are 5/8 of an inch thick. They're actually a plexiglass glass laminate that are put together, and then they caulk them in with a Dow Corning caulking that even 25 years to today still has tremendous amounts of elasticity and almost looks like it's brand new in some cases. So it's done exceptionally well, and that's how they achieved that leak rate. Now, I think this is when probably I began to hear about it. Most of you who knew anything about the project, this is when most of us, it sort of became on our radar screen. So you had these 8 people who were going to be sealed inside this facility, and they were going to live in there, and they were going to be solely dependent on everything that goes on. And I wish I would have brought an image, and I'll embarrass him now, but so we've got a celebrity in the room, Tilak Mahato. So he was part of the second group who was sealed inside. But the first group of 8 were sealed inside, and the intent was to go in for 2 years and see what would happen. Now, you think about it. So they bottled up everything that they could. They have a rainforest, an ocean, a savanna, a marsh, a desert. They have an agricultural area, and you're going to see some of those here in a minute. And the hope is that everything was going to work. So you had the critics saying that it was going to turn into a big ball of slime. You had others saying that it would never work and everything would die. But for the most part, it worked really well. But as often happens, it's our personal dynamics that get in the way of things. So these social and psychological challenges, and one of the Biosphereans, Linda Lay and Jane Tabor, were telling me that they had hoped the sheer size of the facility would mitigate a lot of these social and psychological challenges that had been so well documented by NASA and others, by the sheer size. And what she said is that, in her opinion, it only delayed the onset of those by about 6 months. So they split into 2 groups. They couldn't get along. But nonetheless, it captivated the world, much like sort of The Martian had reinvigorated the general public's interest in space travel and things. And so there was tremendous amounts of media hype associated with it. They were inside, but they had some real challenges. So when they built the rainforest and the agricultural areas, they started out with an extremely rich soil to support this intensive growth that was going to happen over the duration of the project, recognizing that they weren't going to be able to add amendments to support that. So they couldn't go down, for example, to the Amazon basin and truck up soil. They couldn't go to Georgia, which the soil scientists said would have probably been the next best thing, and bring over. So they had to use local amendments. They went to a cattle pond, and then they mixed that together and created a large homogeneous mass. But what that ended up doing is, yes, supporting a lot of plant growth and agricultural growth, and this is their agricultural area, which is about a ½ acre of their space, but it also promoted tremendous amounts of microbial activity. And so now you've got a volume. Remember, we only lose about 10% of it annually. It can't go anywhere. You've got a relatively young group of plants that were just put inside there and planted, and you had these people that were sealed in. There was no test run. The engineers were going out the back door, the Biosphereans were coming in the front door, and they were hoping everything was going to work. You get an overhead view. This is the rainforest, so if you were to look inside, and again, if you were to go there today, you would not be able to see from this end down to the other because the plants have all exceeded, in many cases, the height of the facility, and we have to go in and periodically trim them to keep them. This is the desert area. That geodesic dome on the outside that you can see is the lung, and we'll talk a little bit about that in a moment. And underneath all of that, so you think about all these ecosystem services that are needed to keep things going that we take for granted outside, they had to recreate inside of it. So Biosphere, a lot of people have used the analogy, it's like a ship. So you have all of this mechanical equipment resides in a basement-like orientation to support all of the plant growth that's located above it. Well, what really sort of ended up being the demise, in my opinion, of the Biosphereans inside was, sure, they couldn't get along. They had some challenges with that. Food production was not where they wanted it to be. And so, at least for that first group, they were okay on nutrients, but they were very calorie limited when they were inside. But this was really the crux. So what ended up happening is they sealed that up. And again, remember, they were inside for about 2 years. So a little over halfway through, you had this very dramatic decrease and gradual downward trend in oxygen to around 14.2%. Well, the medical doctor who was part of the team made a decision because he couldn't read his notes and do a few other things, said we need to add oxygen in. So they added it in. But rather than treating it like an experiment, they tried to hide that fact. They didn't even tell the person who was their press secretary at the time. So you can see here, they added oxygen back into the system. Well, again, because there was controversy associated around the project, all of this media hype that was there, when they added it back in, they didn't disclose that, and so they weren't really as forthcoming as some people would have hoped. And then they had to recant their position. They lost all credibility with the scientific, with the public, and with the media. But nonetheless, they were able to stay inside for 2 years. But what this did is it got some folks from Columbia University interested, because as the oxygen went down and went back up, and they were recording all of this, carbon dioxide was equally going up in the atmosphere. And so, but that balance was not where it should have been. So the equations didn't equal. And so a guy by the name of Wally Broker and a graduate student of his, Jeff Severinghouse, they were brought in to figure out what was going on inside the system. And so that led them to say, you know what, we think we can do some really unique things inside Biosphere that we can't do anywhere else. We don't think that it should be used for space colonization and travel, but as a large Earth systems laboratory, we don't know of anything else like it in the world. And what they had found is this guy, Jeff Severinghouse, who has gone on to get his PhD and really well recognized with paleoclimate reconstruction and using ice cores and CO2 trapped in them to recreate that, is his dad is a renowned anesthesiologist, and they couldn't figure out where that CO2 was going because it should have actually been higher than it was. And what they found, because of that mass amount of concrete that was used to build the structure, the calcium hydroxide was reacting with the CO2, becoming calcium carbonate. So it was stored in the concrete. They went in and cored it, and there was a paper that came out as a result of it. But again, this started to get the folks at Columbia very interested. They have a division that they call the Earth Institute. So they came in and they assumed management of the facility. But again, that first group of 8 went inside. They did make it 2 years. Another group, actually TLOC was a part of, they went in for about 6 months. And then there was this strategic shift to move away from people living inside to more of an Earth system science laboratory. So Columbia University came in officially in 1996, and they began to retool the facility. How can we use this for research? Their focus was really looking at CO2 fertilization. So, you know, we know that it's going up in the atmosphere, and how are ecosystems going to respond as a whole? And they thought that Biosphere offered a unique opportunity because of the sealed nature of it and the control capabilities to look at how our rainforest, how our agricultural area, how our ocean are going to potentially respond to varying levels of CO2, and what were those implications for systems as a whole. But in order for them to first do it, they had to break apart essentially this facility that's one big unit. So not physically, but we created barriers inside. So this area here is the habitat. This is where the Biosphereans lived. We've converted this now to office, laboratory, and classroom space. This back area that's green, or it's lit up, is the former agricultural space. And so we grew some agricultural crops, and then towards the latter part of Columbia's tenure, we grew an agroforestry species, a cottonwood, Populus deltoides in there. And this is that wilderness area. So that's where we have the rainforest, ocean, savanna, marsh, and desert. So these are all three distinct areas, and we're separated so that we can conduct experiments in one and minimize the impact that the adjacent areas are going to have on it. Within the wilderness area, we separated or partitioned off the rainforest. So originally these were all interconnected, so we've got a barrier. So now you can independently, for the most part, control conditions in the rainforest area. And independent of the savanna, sort of this ocean area. So these next three areas, the savanna, the ocean, and the marsh, which the marsh left, this is what we call an ocean corridor. So we can't do a lot to separate things in there. And then we can put up a barrier to separate the desert. And so these were the areas where we focused from a research perspective. Again, just gives you a depiction of what we were just looking at on the map. You're looking at a facility, again, that's about 3 football fields in length, 144 feet on the side in the rainforest, 28 meters in the center. The ocean itself is a little over 1 million gallons. And this was the first biome, I think, that had the greatest amount of success in the system as a whole and received the most notoriety for the research. So we saw that CO2 graph with carbon dioxide going up in the Earth's atmosphere, and we've all heard the debates about it potentially affecting our climate and how it's going to affect it. But we know that CO2 is a gas that's soluble in water. And we know that when you add it to water, that it's going to change the pH of that. So one of the first experiments that they did is they took our ocean and they chemically manipulated the water content in the system to simulate a pre-industrial level or an industrial. Then they simulated a present-day level, and then they simulated a future level. And in this ocean system, in addition to the fish and the other microorganisms, they had hard corals growing in it. And so what they were particularly interested in is how are these corals' calcification rates being influenced by increasing CO2. And so what they were able to find is that as CO2 goes up, pH goes down, coral growth slows. And so we commonly hear that today as ocean acidification. In fact, last Friday I had a gentleman who is from one of the world's largest shellfish producers and farmers. He was at Biosphere, and he was telling me that they have a big dilemma because they were finding that their larval state, at about 10 days of their 10-day growth in their larvals, they were all dying, and it was because just a slight change in pH. Now, they can buffer it in the tanks, but what does that mean for the open ocean? Because they lost 90% of their larval, only 10% survived. So if they're seeing that in a tank and it was just a slight pH change that was the driver of that, we know, again, that as CO2 increases the atmosphere, again, it dissolves into it, creates carbonic acid. It reacts with carbonate to become bicarbonate, and now it's not in a readily uptakeable form for things that calcify, and you drive down that pH, which now not only does it impact corals, but other organisms that depend on either pH and that balanced for their next stage in life, or need it to be at a certain level from a nutrient concentration to build their hard calcareous structure. The next area where we did a lot of research in is the rainforest. So remember, it's separated. They started out with well over 400 species in here, and they knew when they built Biosphere as a whole, they were going to pack in as much biodiversity as they could. And the hope was that everything's going to, we're going to lose some species. And their hope was that at the end, when things stabilize, that you would have a relatively diverse community. So today, we just had a graduate student who finished it up, a survey a couple years ago, and what he found is that we started out with a little over 400. Today, we have 98 species. But if you look at the curve, that species loss was in those early years, and in these out years, we get virtually no loss. So we've got a relatively stable community. So now what that allows us to do is we can begin to go in there and perturb the system, and we'll look at how it responds, and that response that we see, we can attribute to a system level response and not because it's going through some type of a stabilization state. And so some of the things that we did is, again, how much carbon dioxide, or what's the net carbon exchange in forests, and how is it going to be impacted as global CO2 increases? And are forests sort of a mitigating component or sponge, if you want to think of it, for these systems? And what we found is a level that we could reach. Today we're about 400 parts per million. When I started working at Biosphere, which was roughly 20 years ago, we were 365. We continue at our same rate of increase. We could double to around 800 in maybe 80 to 100 years. We could reach 1,200 in maybe about 150 years. And so, if we're going to use forest and globally model these global circulation budgets and say that forest and others are going to take up a certain amount, we know that there's a limitation. And what we were able to use Biosphere to do is there was a model referred to as the big leaf model, and we were able to confirm that what it predicted, sort of a saturation at around 600 parts per million, we were able to show that that same trend held true in our rainforest when we ran those simulations or those conditions through our system. Another thing we did is, there's a lot of predictions that the tropics are going to get warmer and drier, and what's that mean for these systems? And so in our canopy, the upper reaches, are roughly 90 feet. It gets very warm during the summer. It's cooler during the winter. So a guy from the Carnegie Institute by the name of Joe Barry over at Stanford, he was really interested in, are these plants adapting their thermal tolerance? And in particular, not seedlings that are grown in different temperatures, but adult species. Are they up and down regulating? And what we found is that these upper canopy plants during the summer would actually upregulate their thermal tolerance by as much as 2.5 degrees C, and then they would downregulate it during the summer or during the winter. And so we did that over several seasons and found that same theme consistent. And that gives you some indication that, as we know, that there are some adaptive characteristics. And in particular, the big questions that come into play is, these adult species that are out there, how resilient are they going to be? And where are those thresholds? Well, Columbia developed and really, if you want to refer to it, kicked the tires of the facility. They did a lot of very interesting things, but they had an administrative change. So the president of the university, he left. A guy that probably most of us have heard the name one way or another, a guy by the name of Michael Crow, who was a vice provost at the time. Those were our champions. So Michael Crow went on to be president of Arizona State University and is still. But when these two individuals left, there was a new administration came in. There was a refocusing of priorities and purposes for Columbia University and the Earth Institute. And so they decided that they didn't want to continue to run Biosphere 2. So they pulled back, and the facility went into a period of time where we were looking at highest and best use. So the person who financed the project received it back, and a group of us went to the National Academy of Sciences. There was NASA, DOE, folks from various universities, and a representative from the University of Arizona, a guy by the name of Joaquin Ruiz, and some others were there. And the notion was, how could we use Biosphere 2 experimentally? Was it still useful? And was there interest out there? And so the consensus was yes, everybody had an interest in using the facility, but nobody had a nickel to run it. And it was difficult to raise the funds to run something as large as this facility was. So what ended up happening is we started discussions with Joaquin, and he went back to his team. So Joaquin Ruiz is the dean of the College of Science, and he went back and said, okay guys, we've got an opportunity. Are you interested in working inside Biosphere 2? Yes. Now, the questions that you can potentially address in the facility, can we do them elsewhere? Because if we can answer those questions elsewhere, there's no reason to get involved with a facility like this. He might still be interested from a public outreach standpoint, but from a research perspective, it's a pricey venture and road to go down. They said, yes, we feel with it because of the large space, the control capabilities, that we can do things inside Biosphere 2 that would be difficult, if not impossible, to do anywhere else. Now, this is a Large Hadron Collider. So you think about the investment that we've put into to figure out, what is an atom. We have large collaborative international efforts. This is a giant Magellan telescope. We use this to look, and we're building more and more sophisticated telescopes every day, some of which, actually the most sophisticated mirrors are built right here at the University of Arizona. We're putting telescopes into space, and we're looking to the ends of the universe, or trying to. But for the earth sciences, you know, we're either working in the lab in many cases on a benchtop that's this big, maybe a growth chamber, or we're stepping outside in the natural environment. And so there isn't this intermediate scale that allows us to begin to try to address some of these large system level questions and these mechanisms that tie things together. Now, one of the mistakes, in my opinion, that Columbia University made is that those systems that you saw some of the videos of, is that they tried to refer to them as analogs. And so it's really important to know that although Biosphere was designed to recreate, in part, Earth, none of those systems are true analogs. And if you try to say that they're an actual analog to an equatorial rainforest that you find in Brazil, there's just no way that you can recreate nature. Okay, and so probably one of the most important things that early group, in my opinion, or that I think that we learned, is how little we truly understand Earth systems as a whole. Because there were a lot of things that happened contrary to what many experts felt were going to happen. But the one consensus is that you take leaf level measurements and how do you scale them up to globally? And we know that our models fall short, but Biosphere is a tool that offers an opportunity at an intermediate step to begin to get at some of these fundamental mechanisms that tie in soil, plant, and atmosphere interactions. Again, Biosphere doesn't have a large mammal population inside, so we don't have large primates in the rainforest. You know, there are a few birds, but most of them are local birds that happen to get in. So there is a food web, and there is significant diversity inside the system, but at the same time, it's not as diverse as that region that you'd find, for example, down in Brazil, or that you would find as you move down the Baja Coast, or if you're on a Caribbean reef environment off the Yucatan Peninsula. And for the University of Arizona, this was really a unique opportunity. So they assumed management in 2007, but it was a management agreement, so it wasn't actual ownership. And the notion is what can we do inside here that we couldn't do anywhere else? And they didn't have the caveat. So Columbia's caveat is when they did their work inside Biosphere, everything that they did, they had to be able to give the facility back to the owner in its original state. So you couldn't sort of drastically change things inside. That was not the condition or the constraints that the University of Arizona had. So Joaquin and his team got together with a number of different folks, and they spent 2 years sort of figuring out, okay, how can we use Biosphere 2? What areas do we think are most useful? And what are those questions that we would like to address that are considered sort of these grand societal questions? And the notion came up is this idea of water. Okay, so we know it rains in the mountains, but how much of that water ends up downstream for societal needs? And how does land use change and changing climate impact that? And arguably, it's probably the single most important resource in my opinion, that's going to be impacted by future climate change. So we see it today. There's either too little water in California, or there's too much water in Texas and elsewhere, in Louisiana. It's never sort of this happy medium. And how is that resource going to continue to be changed over time? Hydrologists will tell you, so Peter Troke, who's our director of research, will tell you that a lot of what they know was developed in the 19th and 20th century in labs and columns. And we still use those same things. But then when you step to the outside world, we know that all of those relationships don't necessarily hold true. So where is an opportunity for us to build something to be able to look at some of these fundamental processes, and not so much how water moves over the landscape, but how does it move through the landscape? How does that landscape evolve over time? And then how does the addition of biology change the behavior of water? Well, that's something that's very difficult to do outside because they're already coupled. The physical and biological world are stitched together, and they muck up things, and it's difficult to sort of tease apart what's going on. Well, we had an opportunity to take those processes, completely renovate this area over here on the left is the agricultural space, get rid of all of those agricultural crops, take all the soil that was put in there out, and build 3 large structures, of which the previous slide showed a gentleman standing on those. So what those are convergent hill slopes. And it spent 2 years with a group, an interdisciplinary group, who came up with the design questions that ultimately led to the building of these structures. They have about 1 meter's worth of basalt, and we'll talk about the significance of why they chose basalt. And they were able to heavily instrument them inside of that. And so the thing that we have is we've got 3 of them inside these bays. And one of the notions is that the physical scientist said, okay, we can get away with 2. The ecologist said, no, we've got to have 3. So we had to build and utilize all three, which increased our budget significantly. But it was something that was critical because even though right now if you were to go out and look at these structures and see them, they're relatively void of any life. But the hope is in about a year and a half, we're going to put plants on them. But today what we're doing is we want to understand what are the physical characteristics of these systems, how does water move through them, and how similar or dissimilar are these three slopes. So when they built these structures inside Biosphere 2, and these are some of the sensors that are embedded in the soil, the notion is, how does life start? Because if you look at our system now, we've got a microbiologist, and she's looked at the soil. It's not sterile, but the diversity is very, very limited. And so now the interest is when we start with something that's relatively sterile, how is that system going to evolve over time and develop? What happens to resources within that system, and how are they cycled? And then ultimately, because we've got three of them, because we can divide them off, we can perturb them. So we can change temperature, we can change moisture, we can change carbon dioxide at some point down the road. We've been slowly adding instruments to this system. So really, we just completed construction. It took us the last year to bring them all up to the same state because when we built the facility, we had to work our way or paint our way out of a corner. Okay, so we couldn't take the top off of Biosphere and build them inside this. We had to build every piece of steel in place, and we had to bring it through an opening that was roughly 10 feet wide by 12 feet tall. And so in order to do that, you had to start with the furthest east, build it, put everything in, go to the middle, and then go to the final western one. Well, what that allowed us to do is do some initial testing on that first one while the others were being built. But it also meant that once we got all of them completed, the instrument were done, we needed to bring them all up to the same state. So we spent about the last year making sure that they've all received exactly the same amount of water, that we understand their similarities and differences, and now we're ready to start a new set of experiments. But along with that new set of experiments is that we've been very fortunate to be able to bring in some instrumentation and put it in place in ways that I don't know of any other place in the world that actually has that. So we've got a new stable isotope facility where we can actually look at, in almost real time, the isotopic signature of not only liquid but also vapor. We put that right inside the facility. We have a postdoc, his name is Till Volkman, and this is his area of expertise. And what we've been, and these are the two laser isotopes that we're going to have running continuously inside the system. But what's even more exciting is now you can look at samples over the surface of your structure or your hill slope. You can look at that which is coming out from and drainage in the structure itself. This is the manifold that they came up with. It's going to draw all of these samples from the three slopes into those systems so you can look at those similarities and differences. Again, what's really exciting is that everything is going to essentially happen real time. So for a long time, you would have to take a sample, run back to the lab, and do the analysis. Now, because of this new technology, we can put that in place. And that instrumentation alone, in and of itself, is a little over $500,000 combined for us. Again, this is where the water drains out of the hill slope, so we can collect that and see. And the significance here is that you can use stable isotopes to trace pathways through the system. And you can look at how does water move through that, when does it begin to break through, and how do those preferential pathways change over time. One of the things that a lot of the folks have been doing there is, so we've embedded the soil with all of those over 1,700 different sensors and samplers. We've got this stable isotope facility, but we also want to look at what's going on over the surface. So we've got a rail that we've hung over the surface, and we've got two different types of cameras that are going to allow us to look at the slope itself and the imagery and look at how that changes over time, in particular when we add vegetation on the system. This is an exterior view of the facility. And those three slopes are in these three bays that you see here. Now, the other area that we've done a lot of focus on is the rainforest. And so much like the Leo area, there's a lot of interest, but the rainforest has been growing for 25 years. So there wasn't necessarily an interest in going in and completely changing the composition and removing. Let's see what we can do and utilize the facility as it currently stands because we've got a relatively stable system. Remember, we're not losing a lot of species. We don't see a lot of change, and we've already had some significant research outcomes. So one of the things we did first is what's going on below the surface. So this is a soil observation pit, and we put 3 of these inside the rainforest. In conjunction with those, we put instrumentation towers overhead so we can look at what's going on at the surface as well as what's going on as you move your way through the canopy inside the facility. And the big questions we're asking in particular today is, what happens when this system goes through a drought? So the tropics are going to get warmer and drier, but it's really hard to go down to the Amazon basin and turn the spigot off. We can do that inside Biosphere 2. And we can subject the system to an extended drought of our choosing. Now, we want to push the system, but we don't want to push it over the cliff because we want to be able to then come back and do some experiments. So this gives you a layout, shows you some of the vertical profiles that we have in the system. These red boxes down below here are those soil observation pits that allow us to look at, again, what's going on subsurfacely. Probably the most important thing to remember about Biosphere today, in addition to that those systems are analog, is secondarily is that we're no longer sealed. So much like the greenhouses that you have out here, that Gene and his team have, we move air in and out. And for us, if we can quantify the volume of air that's coming in and its makeup, and we know what's going out, its rate, and how it changes, we can determine fluxes. So we can mass balance our systems. And that's what we're doing for things like methane, nitrous oxide, carbon dioxide, and water in particular. The other really interesting thing that it's difficult to do outside, if not impossible, is we can isotopically label our rainforest soil and water and look at how the system responds. So Joost Van Haren, he's our faculty who's overseeing the rainforest. And there's an interest that as we dry this system down and we really begin to stress those trees, where do they get their water from? And when do they begin to take that up in the roots? Well, how do you know that? Well, essentially what we can do is we can use a label. It's almost, if you want to think of it, if you're not familiar with it, like a dye. We can actually inject it in these tubes here because that's where the water would have otherwise drained out. We've already dried the soil out. Now we're going to inject it, and then what we're going to do is the plants are going to allocate their resources to the roots. They're going to seek out where that water is, and they're going to begin to take that up to support their existence because they're in this drought scenario. And we can actually begin to detect that in the systems and look at where does that end up showing up in the system. Something that would be virtually impossible to do on the outside. Again, you saw some footage, but you can see how much has grown in the system. The other important component is the LEO project, the Landscape Evolution Observatory, those large hill slopes. That's coupled to a national project called the CZO project, the Critical Zone Observatory. So this is a network of watersheds that have been instrumented in somewhat a similar fashion, and Peter Troke for a period of time was one of the PIs on that project, and there's one right here in the Catalina Mountains. There's one over in California, there was one in New Mexico in the Jemez Basin, and then I don't know exactly where all the other locations are, but there's a few others as you move further east. Joost not only spends time here, but he spends time in the Amazon Basin as well, and so it's important for us to be able to couple and tie our research, not only what we're doing inside, what's going on in the lab, but also what's actually going on out in the field. Because Biosphere is this intermediate step that allows us to understand fundamental mechanisms and some ecosystem processes, but again, there's no replacement for the real world. So that's the conclusion. I'm happy to answer any questions that you may have, whether it goes back into the historical days or we talk about more about what we're doing today. Jack, start us off.
I'm thrilled, and this is a fantastic presentation. One of the things that comes to mind is the comparative activities between various units, and here the study that we have going on with tomatoes and cucumbers, et cetera, et cetera. I'm under the impression that with some work that's going on with Raytheon at this point, we have an ability to look at the behavior of root systems under the soil through opaque soil. And I'm wondering if we're going to be looking at the changes that roots undergo in your laboratory up there and compare it to what Gene has, where the root systems are in an aquatic environment?
We've done a little bit of that. The soil observation chambers that we put in allow us to look at roots below and subsurfacely. We have wired mesh glass, so we can look at sort of how much growth occurs over certain periods of time. But for us to completely excavate and completely look at root structures as a whole, that's something that we're not going to be able to do in our system. Now, Dragos in some of his work has looked at root structure, but again, we can do things on a sort of smaller scale where you can stick a tube in, run a camera down, take images over time, and look at how those types of things change. But again, once you get below that subsurface, it's still a black box, and being able to monitor those conditions in a way that is truly meaningful oftentimes means you have to do some destructive sampling. And at least for us, for many of our systems, that's not an option. Now, we do some smaller, what we call opportunistic experiments, where we have that ability to do so, where we can completely excavate the system or remove the material and look at, as much as we can, the soil and the root structure as a whole. But for us, I think, unless there's a new technique that I'm not familiar with, that's, yeah, well, I think it's good. Yes, I mean, we would definitely welcome that type of partnership, but I'm not familiar with those techniques and how that would come into play until we explored them.
I think it needs to be developed, is what we're saying.
Yes, but it has the potential, right, Jack?
We'll keep working on
it. Excuse me. In a normal situation right on the earth now, we know the plants grow, make seeds, the seeds drop. In order for them to survive, they have to have seeds. How do you take care of that in your environment?
Right. No, that's a great question. And so early on, yes. So the question is that how do we deal with new recruitment or seed germination inside Biosphere 2? And the answer is we don't, to be frankly honest with you. So they had hoped early on, as I understand it, as part of the design criteria, to have a number of species that would act as pollinators. So they had introduced bees. They quickly died off. They had hoped to have hummingbirds and some other insects that would have potentially aided in that. But it ended up not working out as well as they had hoped. And so there were a few insects that became very dominant inside, and so we do get, some passive pollination. There were efforts made in the agricultural area. TLOC and others would actually hand pollinate a lot of those species to get things germinating. From time to time, we do get recruitment, for example, in the rainforest and other areas, but it is believed to be primarily sort of accidental or a passive pollination rather than an active pollination. And so as a result, over time, you're right, that system, because we don't have that continuous recruitment, we're going to reach a certain level where we'll have to make a conscious effort to either introduce species or step in and actively try to pollinate if we're going to continue to see those species reproduce inside, because we get very little to none of that currently.
Thank you. All right, so a couple things. One, what do you? I know you mentioned earlier that you don't want to excavate much in the rainforest area, but what do you think about excavating a certain area and then placing sensors along, say, maybe every foot or whatever increment you decide, and then making a matrix to where you put soil on top of that, and then so each sensor is placed by like one by one by one dimensions, and then placing a tree or plant or whatever you want to test the root system of and the development of that, and then see how that interacts with those sensors. So one thing. And then the second thing is, well, what's the annual operation cost for the facility?
Two very different questions. Yeah. Okay, so we'll start with the first question, and that's, that is a great experimental design. And actually, LEO, the Landscape Evolution Observatory, is set up in a manner that's going to allow us to do that. So currently, I don't know of any plans to do that type of an arrangement in the rainforest. But LEO, because of the network and the distribution of the sensors, yes, currently we don't have plants on those basalt soils. But the hope is that, again, in about a year and a half, we will begin to vegetate those. And the community is yet to be determined. There's several discussions out there. But then we will actually have an opportunity, because those sensors are already in place. You're going to have a soil that's evolved to look at how do these plants grow in that system. The sensors that were put in LEO, though, are ones that were designed to last a very long time, or at least that's the hope, because we knew that we weren't going to want to go in there and necessarily excavate in the fashion that you had described. So we'll have an opportunity to get, I think, a lot of very interesting data from that particular system. There were some early experiments where they removed soils in that former agricultural area and replaced them with a C4 soil, and they did that exact where they would take out an area and they isolated it. But at least currently under the university's management, we haven't done anything like that. And I think, to be honest with you, we're still trying to understand what we have in the functionality. And those soil observation pits, we did extensive excavation and completely removed a large portion of soil from that area. But you're right, we didn't re-instrument it, put the soil back in, and then put a plant on top. But the hope is that the selection was in an area that gave us the greatest opportunity to look at roots and some of the things that they're doing. And so they specifically selected those sites next to some of our dominant canopy species in hopes of being able to achieve, in part, what you alluded to. Jumping to the budget. So that's a great question, and it's one that we often get. So we talked about the estimated cost of building the facility. Today we have an annual operating budget of about $5 million. For us, though, we're unique in the sense that we don't have a state line. Okay, so we don't get any state funding through traditional tax lines like a lot of other, like some university entities do. We pay for our own utility bills. We pay for our own gas. We pay for all of our own maintenance. And so all of that comes out of a philanthropic gift that was given to the University of Arizona to support the operations and maintenance of Biosphere 2 by Ed Bass, the person who originally funded it. And then the other is and why I want to encourage all of you to come up and visit Biosphere 2, is the revenue that we generate. So we're an auxiliary unit that we get from tourists coming in. And so on an annual basis, we get about 95,000 visitors. And then we have a small conference center up there. So we have overnight accommodations where we can host conferences. And so it's the combination of these three things that currently provide our operational expenses and allow us to do the maintenance and pay the staff that we have. Now, above that, our team, and in particular Peter Troke and the others, have been very successful at recently getting a number of grants through the National Science Foundation in particular to support research, in particular on LEO, the Landscape Evolution Observatory. So that's above and beyond, and research dollars are great, and it helps to confirm, especially when we win one of these very competitive grants, that we're on the right track and there's a recognition of what we're doing and there's an interest in that, but it doesn't pay for the O&M costs. So it's going to pay for the grad student. It's going to pay for Gene and his team to buy instruments and do things like some of the laser spectrometers that we saw, but it's not going to pay for me to go fix one of those air handlers that allows us to circulate and maintain the conditions inside. So both of those are vitally important for our existence, but sort of two separate lines if you want to think of them.
I first wanted to congratulate you for the presentation. It was a great vision that you have introduced us to. Now, at Earth scale, you have the Earth is run through these cycles, right? You have a hydrological cycle, we have a nutrient cycle that supports life. Both of them have like an air cycle, right? And we know that Biosphere has this concept incorporated when it was first built. Is there any monitoring of the nutrient cycle that happens or had happened in the Biosphere, or regarding the water cycles?
So currently, they do a lot of that with LEO. So LEO, so there was a recognition early on that we would have liked to have had that original group do a lot more scientific monitoring and documentation. But again, and I wasn't there at that time, I came in shortly after that, but as we understand it, a lot of their emphasis was put on to survival and getting along, and so a lot of these scientific aspirations that they had, some of which were spotty, and there is a great set of publications that helps to document in part some of those early conditions in a journal called Ecological Engineering. I'm happy to share that group because there was a special issue. So it did document in part some of those nutrient levels in the rainforest, some of the decomposition rates, the nutrient levels and soil composition in the agricultural area, the water conditions in some of the systems, freshwater and saltwater. But as far as a continuously monitoring system, no, that wasn't part, or that's not data that I've seen that's actually been published other than what I've just referenced. Now, with LEO, there has been painstakingly efforts put into developing systems that's going to allow that research team to collect samples from a wide range of locations within different soil profiles so that they can do these types of analysis and have a better understanding of what that matrix looks like and how it changes over time, especially as this basaltic material begins to weather more significantly over the life of the project. Does that answer your question?
Yeah. And what about water? Has the water fluxes been monitored?
So there's a couple of publications that look at the water budget, but again, most of those studies were intermittently. So they were for short periods of time. So there was a campaign. They would try to close the water budget under a certain set of conditions. But as far as continuously monitoring, no, that's one of the areas that we wish they would have done a much better job. And in some cases, we just don't have a complete picture as we would have liked from sort of the beginning to where we are today.
Hi, John.
Hey, girl.
Okay. Like I understood, if I understood correctly, you're focusing right now for two biomes, right?
Correct.
Agriculture and rainforest. What about another biomes?
So that's a great question. So another area that we're beginning to see a lot of interest, and we had a lot of interest early on, but we had a tragedy among our team where we had Dr. Rafe Sagrin, and this is the ocean. So the ocean received a lot of interest and success early on. There were some other follow-up studies that folks did, but then when the university came in, you're right, we focused on the agricultural area. There's been a focus on the rainforest, but concurrently what was going on was a focus on the ocean itself and how could we potentially retool this system in a way that was going to allow us to use it to a greater scientific capabilities. And so a gentleman by the name of Rafe Sagrin began down that pathway of looking at redesigning what would that look like, what were those types of questions that we felt that this system potentially would be well suited for. Unfortunately, a little less than a year ago, he was tragically killed in an accident near Biosphere, and so that sort of stopped us a little bit. Now we've jump-started that again, and we've brought on, maybe some of you know her, but her name is Julie Cole, and so she's at the University of Arizona, and she's going to help head up our marine science program, and she's doing exactly that. She's convening experts in a team similar to what we did for LEO of interdisciplinary folks to look at what do we need to do to the ocean to potentially increase its scientific usability. What are those types of questions that potentially could be answered inside Biosphere that you couldn't do elsewhere? And then how do you need to redesign it? Does that mean you need to drain all the water and start from scratch? Does that mean you need to change simply the instrumentation in the system? What does that actually look like? So we're in the process of just beginning to restart that again. But those, the focus, primarily is LEO as the institutional, the rainforest, and then the ocean.
I'll put in a plug here for Rob Wing, who's a wonderful guy and has done great things with genomics of rice. And only recently did I discover that closed environment agriculture has the largest production of rice on this campus, because in a desert, who the heck would ever grow rice? But the issue of how to get the greatest amount of knowledge out of Gene's laboratories and your laboratory with the right monitoring of the various variables that go into better forms of rice with greater resistivity to disease and greater nutritional value, I see a very good potential. Are you going to have square foot operations that you'll be able to outsource to various other people within your agricultural faculty that will be able to do studies of some nature that get things ready for the fields of the Philippines from here in Tucson?
So the short answer is yes, and we're just beginning to explore some of those, and we're looking at, you know, the overlap that we have between Gene and his team, some of the vacant space that we have at Biosphere 2, and is it suitable with some modifications to outfit and leverage, in fact, some of the folks that he just had here for, you know, doing some type of commercial venture with indoor agricultural systems. But we're really in its infancy of beginning to explore those and looking at those options. But yes, the notion is that the belief is that we've only scratched the surface on the usability from a research perspective of Biosphere. And again, the option is, you know, for example, in the desert, and if you were here for Dragos's presentation where he did, you know, it's an area that, at least in my opinion, we feel like we're underutilizing. And so there isn't limitations. So if somebody comes forward and say, if you cleared this out or if you changed it in this way, I could do X, and if they had the dollars to do so, it's my understanding and it's my belief that the Biosphere 2 team and board would be very receptive to those types of things. And so what we're hoping is to continue to get more and more people involved to come up with ideas that we had never even considered using, either using the systems as they currently are or completely modifying the space in a particular fashion like we did with Leo and building something that's never been done.
I got a question. You got a ghost town beyond the biosphere of greenhouses and stuff. What's going on with that?
That's a great question. So this goes back to, so when the University of Arizona assumed management and then ultimately ownership of the facility, it was a very small footprint relative to the outside property holding. So the group who donated Biosphere to us, they hold about 1,600 acres that surround Biosphere 2. We were allocated or donated approximately 40 acres. And if you want to think of it, it's a donut hole inside of this larger property holding. And so the group who owns that, they are looking to, in part, develop some of their surrounding properties, in particular that which is adjacent to and butts up against Saddlebrooks, because Saddlebrook's just to the south of us. As far as those other facilities, so there's, of that 1,600 acres, if you lump Biosphere in there, you're looking at about 250 acres that's developed or has structures similar to what Phil had referenced. And so I don't know what we're going to do with those or what they're going to do with those. You know, there's been a wide range of discussions of repurposing those, creating some type of industrial tech park type area, developing it and putting some type of a more formal hotel accommodations that would be able to support conferences, to renovating the greenhouses and, you know, doing similar type things or offering opportunities to, you know, what's happening here on site. So there's been a wide range of possibilities put out there, but nothing formally has come up yet. Those are privately held, and at least I don't see those coming over or being donated to the university for a while, but who knows? It's always a possibility.
Other questions? 3 hands go up. Wow. Okay, Sean?
Had a question about specifically with respect to the LEO project as far as the water distribution system.
Mm-hmm.
I know for LEO it's talking a lot about water movement through the soil. Is there like a rain system or that distributes the water to study that?
Yeah, that's a great question. So, you know, how do we distribute water in LEO? And so first, to understand the water that comes to Biosphere, I'll take a step back and that which is sent to the site. So we get our water from a well source. It's believed to have been deposited during the last glaciation. It's about you know, an 800-foot drop to get that. We pump that out, so it's a groundwater source. It's a different aquifer than what's here in the Tucson Basin, so it sits up into the sort of the Catalina foothills. And that aquifer is drawn upon very little by anybody else other than us, at least for what we can tell. We pump that out. We bring it up, a couple staging tanks, and then we put it inside Biosphere. We first run it through a reverse osmosis process, and then we put it into large holding tanks that sit in the basement of LEO. From there, through a series of pumps, it's pumped up to the slopes, and then on the perimeter of each slope are sprinklers. And so there's a configuration of sprinkler heads that run at different rates to allow the research team to dictate whether they give a very nominal or a very heavy rain on the slope. Now, the notion is that when we put LEO in place, once we established and started the experiments, we didn't want anybody touching the soil surface. So we have a gantry system that allows us to access it. But the other thing we didn't want to have happen is we didn't want to have an overhead rain system that potentially had a PVC pipe or other that would rupture and then create a gully that would be difficult to reproduce on the other two slopes. So we spent quite a bit of time. There were a couple of graduate students who set up a number of different sprinkler configurations on the ground, a similar footprint, and looked at different types of heads that were going to give us the greatest amount of distribution and uniformity. And what we've had to come up with is we have a couple of different circuits on the LEO slope and these perimeter sprinkler heads. We can run those in a certain combination to give us the greatest uniformity distribution over the slope itself. And that was fundamental as one of the keys, because we wanted to be able to try, again, to have equal water over the slopes and have minimal dry or wetter spots in areas that would potentially contribute to different physical characteristics that might be difficult to detect in the sensing network. So we put it on the perimeter, and the reason we put it on the perimeter is we don't want things overhead that would potentially break over time and create other problems.
All right. Couple questions again. I promise these two are very similar, okay, no or problem. More similar than the other two. So first, have you done any bacterial analysis? You mentioned that there was a lot of different bacteria, and did you introduce any of that bacteria, or is it all okay?
No. So that's a great question. The basalt material came from northern Arizona. And we didn't introduce any bacteria. We knew that inevitably, because we couldn't sterilize it through the transport process and through its native site and through the grinding process to get it to achieve a loamy sand, that inevitably we were going to have some microbial communities in place. We didn't know exactly what we were going to get. We have a postdoc, her name is Aditi, and she is beginning to look at that and characterize it, and then is going to monitor that over time. We did take time zero samples, so we cored it and analyzed those, and then we're also storing those for future analysis, and we're going to continue to do that. But for us, the coring is going to be very limited, because as you could imagine, a project that we anticipate running 10 years, we don't want it to end up looking like Swiss cheese. And for every core that we pull out, we've got barrels that we've set up with the same soil that we've tried to water and do the same thing to, so we can plug that hole essentially. So we've done some things, but other folks like Dragos and others have done some things where they've looked at increasing the viability of soil and accelerating the weathering and more susceptible or more optimal for plant growth by adding bacteria and other things, and he can speak about that more than I could.
Right. Second point. So since the ocean, in the ocean, there's a lot of upwelling, and that's how, you know, the sediment and whatnot is carried around. In a, what is it, 25 by, how big is the ocean?
Okay.
Section?
So, yeah, so the ocean actually has a lot of topographical variability to it, and it has a deeper end that's roughly 7 meters, and so you're looking at an area that's you know, about 33 meters long and 11 meters or so wide approximately, and proceeds up into a shallow, like reef-like structure in the center, and then into a shallow lagoon, ultimately up onto a beach. Now we have wave action, but we have relatively uniform mixing in the system. There's no upwelling or different variations or thermoclines that are created in the ocean.
Okay, so then, say, since carbon dioxide, you were measuring how you know that was being taken up by the water. Just playing devil's advocate here, I mean, how can you say that that can be related to, say, the ocean, where you have a huge, huge body of water and all these other complex currents? And do you ever plan to introduce marine or fish or any of those type of animals?
So there were and there still are fish in the system. There are no longer hard corals, but that was part of the original system. And the study that I referenced early on did have those in place. So at the time that so a couple of questions, and I'll address them. It'll be a little drawn out, but so yes, the system was very diverse to start off with. Inevitably, much like the rainforest, you saw a reduction in diversity over time, just because you have very different relationships that are developed. It's much like an island, if you want to think of it as island ecology. It's artificial. Yes, but with that being said, it's very clearly understood that as you change atmospheric CO2, it's a gas that's going to be soluble. So there's going to be an equilibrium. And yes, we know colder water is going to hold more CO2 than warmer water, but you can very much see, and you can even see today, if you look at historical pH levels to today, there's been a significant drop. And, it's directly attributed to increasing CO2. So in part, if you want to look at a larger scale system, bigger than a traditional aquarium at some of these species, and that sort of interaction in a system that has closer characteristics to an outside system where you've got a complex food web in place. It's not an aquarium in the sense of a traditional aquarium. It's one where the internal nutrient cycling of all of the organisms were dependent on one another. So we weren't going in and feeding the fish. Yes, we'd maintain water temperature. We'd make sure the chemistry was where it needed to be. But unlike an aquarium where you would feed your fish and things, that wasn't happening. So everything was internal to the system. And so that gave you closer comparability to that you have on the outside and why they do. But you're right. What it allowed them to do is to show that there is something going on here, and that we see a direct correlation between increased CO2 and decreased coral calcification, so much so that then that has led to a number of other studies, both in laboratories but also in more traditional marine settings, to be able to continue to investigate exactly what's going on. And they've been able to confirm that that's exactly what's happening.
Now back to Leo and the rainy system. We know it's important to create a homogeneous rain on the hill slope, or not so, depending on your experiment.
Right.
Have you ever explored the idea of printing the rain?
Doing what?
Printing the rain on the hill slope.
Printing.
Printing the rain on the hill slope.
So I'm not familiar with that.
You would have this arm that would go up and down.
Oh, yeah.
Print.
No, that's a great, okay.
Right.
So
as compared to the sprinklers, where you can't control anything.
So we did look at that. And so, what if we would have had, like when you go through a car wash, moves up the slope, rains, goes down. We did actually, that was part of the discussion. And the notion is that we wanted, that Peter and many of the hydrologists felt that it was more important to have the slope simultaneously receive water rather than have it in sort of like this sheet type delivery. In particular because they felt that you could potentially increase the wetting over time in some of these upper areas or lower areas, depending on where you start and how quickly that moves over, that you could then develop flow through the soil that may not have otherwise occurred given this sort of sporadic delivery of water, or even if it's not uniform, but very spot type delivery. So they felt that a more uniform distribution over time would give them a more realistic, even though it's not a perfect rain system in the sense of what you get on the outside, delivery, and then they would be able to more closely look at the behavior of water as it infiltrates into the soil and then ultimately moves down the slope itself as a whole. So yes, they did look at it, and those were some of the reasons, and I'm sure they've even got better explanations than I did.
So my question had to be revised because my colleague over there stole my thunder. But I wanted to return to one of the early comments that you made where you were addressing the issue of pollination and suggested that some birds invaded the facility. So instead of relying just on them, are there any thoughts of introducing some additional insects and bird life into the facility?
So that's a great question. There have been sort of discussions started and stopped with that. I don't know of any currently, but even today, if you were to go inside Biosphere 2, and if you venture in there at night, there is a very diverse, there's relatively speaking, there is a diverse insect community. So if you go in there at night, there are a number of roaches in the system. They were intentionally, well, I know we all laugh, but they were actually intentionally introduced into the system because they're very important to initiate this detritus breakdown. The ants are still present. There are spiders. The few birds that have gotten in, I'm not sure if they're mostly English house sparrows and some cactus wrens and some rock wrens. So I don't think they're doing a lot of pollinating. Just because of their particular behaviors and characteristics. But inevitably, the roaches are moving from flowers to flowers and doing things. So you got to imagine they're carrying pollen grains. The ants, so there is a species of ant called crazy ant. And if you ever visited Biosphere early on, they were everywhere. It's Paradochinus longicornis is this genus and species. But we actually had an entomologist from Columbia. His name was Jim Wetter. He published a couple of papers. But the ants became very dominant inside. They were not intentionally introduced like the roaches, and they outcompeted virtually all of the other insects that were introduced. But there is still a food web and a diversity. I'll get back to that story. But if you dig in the soil, you're going to find mollusks, so you're going to find small snails. You're going to find roundworms and flatworms. We've got a few reptiles in the system. We've got some amphibians. There are some spiders. There are some scorpions, all relatively limited in quantity, but nonetheless we do see them from time to time. But back to the ants were accidentally introduced, and he actually made a correlation common to what we see in a lot of introduced or invasive species where they take over a system, and that's what they did inside Biosphere. And they seem to follow the population dynamic, so it's a classic predator-prey relationship with the roaches, and we believe that they were consuming those. So right now, if you go inside, they're not nearly as abundant, but if the roach population picks up, then I suspect we'll probably see them come up and go down. But this species has shown up on the Hawaiian Islands, this crazy ant, and he made a correlation that maybe over time you could potentially see a system that has a significant decrease in its biodiversity directly attributed to this particular ant species, similar to what he observed inside Biosphere. So they've done good, I think, and have done some pollination. But at least right now, I don't know if there's any discussions looking at adding insects. But I think none of those things are off the table.
That's excellent. No, we're going to have to wrap this up. This is Friday afternoon. John, you just did an excellent job.
Well, thank you all very much.
Please. What an opportunity. What a jewel we have in our backyard here. So this is wonderful. Thank you for your time.
You're welcome. Thank you.
And thank everyone for coming here. And those on the internet, we're sorry we dropped off. We lost I think when you were talking about finances, we went down a little bit. But we came back up.
That's the best time, guys.
The science, all the good science got out there.
Good.
Was there a question?
Yeah, I just want to make a short announcement.
Okay, there is an announcement.
It's a double-headed announcement because Jack mentioned Rod Wing and his contribution to the genome of crop plants. He is going to be given our last seminar at the end of April. So if you want to hear directly from, and he is really a treasure at the university and worldwide, so he'll be talking about the new super crop plants.